let me just talk about how to write the genotype and phenotype ratio for blood types. And so for genotypes, there's a lot of different genotypes for blood types. And so, like, for instance, number one, let me show you how I did number one here. So here's the cross. The first parent is heterozygous for B blood, so that means they have a B gene and an O gene, so big I, IB, little I, and IA, IA. So that's the cross for number one, because it says that the second parent is homozygous for blood group A, which means the two genes are the same. So to get this right, you had to have the setup correctly. Then for the genotype ratio, instead of writing zeros for all the different genotypes, because there's like 6, I think, different genotypes that you can have for blood type. That's a long genotype ratio to write. So you can just write, as long as you write what genotype you're talking about, you can just write the ones that are present. Does that make sense? All right. And there's not a particular order either. Okay. So 2 IA IB to 2 IA little I. And same thing with the phenotype ratio. You can just write the phenotypes that are present as long as you just write what they are. Okay, so for blood type, I just wrote AB. It's type AB, so it's 2 to 2, 2 ABs and 2 A blood. So what this does is tell you that these two parents have a 50% chance of having a kid that's type AB or 50% that's type A. Okay, so that's how that's done. And so what I'm going to do is, for the rest of them, I'm going to read off what the cross is, so you know what the cross is, and then I'll read off the genotype and phenotype ratio. You can check yours, and then at the end, if you have any questions or want to go over any ones, we can. Okay, so number 2, the cross was between IA-IB and IA-IA. All right, and then the genotype ratio, you have 2 IA-IA to 2 IA-IB. Phenotype ratio would be 2 type A's and 2 type ABs. Number 3, the cross was IA-IB with IB little I. And the genotype ratio is 1 IA-IB, 1 IB-IB, 1 IA little I, and 1 IB little I. You can have those in any order. In the phenotype ratio, you should have 1 AB blood, 2 B bloods, and 1 A. Question?
Instead of writing like IB I for like the heterozygous B, can you just write like 1 heterozygous B? Is that like infers that it's an IB and then an I?
For the genotype? Yeah, you should write the actual genotype.
Okay.
Yeah. Number 4, the cross is IA-IB with a little I. And the genotype ratio in the kids would be 2 IA little I to 2 IB little I. And phenotype ratio would be 2 type A blood to 2 type B. 5, the cross is IB little I with little I. And the genotype ratio is 2 IB little I to 2 little I. Phenotype ratio is 2 type B to 2 O. 6, the cross is IA with little I. Genotype ratio is 4 IA little I's to, and the phenotype would be all 4 type A1. 7, the cross is IA-IB with another IA-IB, and so your genotype ratio is 1 IA-IA, 2 IA-IB, and 1 IB-IB, and the phenotype ratio is 1 type A to 2 AB to 1 B. And lastly, 8, new cross is IA little I, crossed with IB little I. Your genotype ratio would be 1 IA-IB, 1 IB little I, 1 IA little I, and 1 little I. And your phenotype ratio is 1 of each type, so 1 AB, 1 B, 1 A, and 1 O. Okay? Any questions on those? No? All right. So with blood type, so sometimes you can use blood type to determine paternity sometimes. For instance, if you had 2 parents that one is a type AB blood, I don't know what's going on over there. One is a type AB blood and one is a type O blood. And if they had a kid that's a type AB blood, is that possible? How many people think no?
Wait, type what?
So these are the two parents,
Oh, okay.
and this is the kid.
Does it have somewhere? I don't know.
How many people think it's not possible? Can somebody tell me why it's not possible? Raise your hand if you can tell me why it's not possible. Allie.
Because the AB would have to be an IA, IB, the O would have to be two I's, and if we across, we would get an IA.
That's right. So this is what the genotypes, you would know without a doubt with the two parents genotypes, because these are the only two, the genotypes they could be. And so, yeah, and so when these two have kids, they can only have either an IA with a little I or an IB with a little I. This parent is always going to give a little I to their kids. And this kid here has an IA and it's an A. It's supposed to be an A. An IA and an IB, which means they had to get an A or B gene from the other parent. All right, and so so that this is not possible. So in some cases you can tell paternity. In this case, usually the mom is not a question because the mom had the kid, but the father is usually what's in question. Sometimes you can use blood type, not all the time, because for instance, if the parent, you know, if sometimes you can have A and B and O, and in this case here, is it possible for an A and a B parent to have an O kid? Yeah, if the A parent is this and the B parent is this, the two little I's can come together. So not always, but sometimes you can, okay? Alrighty, and so that's where we're gonna leave off for the test for tomorrow, okay? So the rest of the hour, what I did with the other 2 hours is we just did an overview, again, like we've done in the past, okay? So, is that okay with you guys? All right, sounds good? Sound like a plan? Righty. All right, so we'll just go through. There's 3 chapters. There's chapter 12, which is mitosis, chapter 13, meiosis, and 14, we're getting into genetics with genetic problems. So chapter 12, let's just go through each one of these. So 12, so the first section, 12.1. The first section had a lot of vocab in it, so a lot of things that are built upon later on in the chapter. So for the vocab, we looked at somatic cells versus gametes. Do you guys remember what the difference are? Yes? All right, so somatic cells, let me put this down so we can actually see it. Somatic cells are any cell besides an egg or a sperm cell, so any other type of body cell. Your gametes are your egg and your sperm cells. And so the first 2 chapters, 12 and 13, are about cell division. We have 2 different types of cell division, mitosis and meiosis. Somatic cells are made by which type of cell division? Mitosis, right? And so I'm going to put here, so somatic cells are made by mitosis. And your gametes are made by meiosis, right? And so that's why we have to have a separate cell division for gametes. Why? What's unique about gametes?
They're haploid.
Yeah, they have they're haploid, which means what? Half the amount of chromosomes. So another way to say this is we have 1 set of chromosomes, or 1 of every type of chromosome as well. Okay, so then in this I introduced chromosomes versus chromatin. Just remember what the difference is with those? Katie? That's exactly right. So chromatin is uncondensed. It's DNA wrapped around histone proteins, but it's more stringy-like. It's not condensed. And then it condenses into a short, stocky little structure called a chromosome when it's going to go through cell division. What phase of the cell cycle do we find chromatin in? Yeah, interphase. Absolutely. So then, when the chromatin replicates and condenses, we end up with replicated chromosomes. And let's put these C-somes for chromosomes. Replicated chromosomes. When we say they're replicated, they have two sides. They kind of look like the letter X. All right, so here's one side, and here's the other side. What are these sides called? Sister chromatids. And what connects them? Yeah, the centromere, which has spots on them called the kinetochore region, which is where the microtubules attach. Absolutely. And so, so replicated chromosomes, these are made out of, so these are two sister chromatids. What's the relationship between the sister chromatids? They're exactly the same. So the DNA making up this chromatid is exactly the same as the exact replica of this DNA making up that sister chromatid. And attached at centromere. And the centromere is right here. And this whole thing is what we just looked at here, a replicated chromosome. So that's one replicated chromosome made out of two sister chromatids attached at the centromere. Okay. And so those are some of the vocabulary in the beginning there. So then section 2 of chapter 12 was about the nitty-gritty about the cell cycle, and part of the cell cycle is mitosis. So cell cycle and the phases of the cell cycle. So I'm going to do it in categories here. So let's talk about what phases comprise the cell cycle. So the cell cycle has two main parts. The cell cycle, remember we looked at it and it looked like a pie graph almost, pie chart. So the first phase is interphase, which is the collective name for the combined, for three subphases that are combined into this interphase. What are the three subphases of interphase? G1, S, and G2. Interphase is not really cell division. The interphase is a cell prepping for cell division. So therefore it's getting ready to divide. So in the G1, the cell grows. What happens during the S phase? DNA replicates, right? The synthesis of DNA, and then the G2 it grows some more. So it's prepping for division. So you need to know what happens in each one of those. In interphase, we just talked about how the DNA is still in the chromatin form at this phase. And so like if I were to draw a cell in the G1 phase, here's the nucleus inside the cell. The DNA is in this long stringy phase that's chromatin. So this is chromatin, tying in what we did up here. So there's two pieces of chromatin here. Let's say that that's the DNA that will make up like one pair of chromosomes. So then when we say S phase, the DNA synthesis or the DNA replicates, you have your original piece of DNA and it's still in chromatin form, but now we replicate it, so this is an exact copy. And then this side, this one also, here's the original, makes an exact copy. And it's still in long stringy form, so it's still chromatin in both of these. It's just replicated here. And it's not until it gets past interphase and starts mitosis that it begins to condense and form into chromosomes. All right? So that's interphase. Sometimes cells can come out of this into what phase? You guys remember? G0 phase. That's the non-dividing phase. And some normal cells do this. What do you guys remember the examples I gave? Liver cells and neurons. All right, part of your nervous system there. Liver cells can be triggered to come out of the G0 phase and divide if damage occurs. So yes, absolutely. And then the second phase of cell cycle here, interphase, the second phase is called the M phase, or mitotic phase. And this actually has two parts, the M phase. The first part is mitosis. Mitosis is also subdivided into smaller subphases, kind of. Do you guys remember the phases of mitosis?
Prophase, chromatophases, metaphase, anaphase,
Prophase, prometaphase, I'm just going to abbreviate, metaphase, anaphase, and telophase. And that's the end of mitosis. All right, so so you guys kind of have to kind of know like the flow of chromosomes through that. All right, so so kind of the general idea of what's happening, the role of the spindle, and so on. I don't have enough time to go through every single phase individually again, but need to know that with that, so that's mitosis. And why do we do? So those are the phases. What's the purpose of mitosis? For what reason do our cells go through mitosis? Growth and repair, absolutely. And what kind of cells go through mitosis? Somatic cells, right? Absolutely. Okay, and so that's mitosis. The second part of the M phase is when, at the end of mitosis, you actually don't have two cells at the end, because telophase is the end of mitosis, and so what you have, if this is the animal cell, this is what you have. It's starting to indent. You have, if this is the animal cell, this is a cleavage furrow, and you have two nuclei with your chromosomes inside, and so on, but it hasn't yet split into two cells. So you, at the end of telophase, you have one big cell that looks like it's starting to divide into two with two nuclei in it. And so the process where it actually splits into two is cytokinesis. So the cell splits into two cells. So that's that moment where it keeps on indenting, kind of cinching into two until you get those two cells. So at the end of mitosis, the process of mitosis, we still have one cell in there. And so really then mitosis, another way to think of this is we could say it's the division of the nucleus, because you end up, that's what you end up, you start out with a cell with one nucleus with 46 chromosomes if it's human. And at the end, you have one big cell with two nuclei, each with 46 chromosomes. And so that's what we really have. It's not until cytokinesis that it splits and you get two cells with 46 chromosomes. So when cytokinesis eventually does occur, and you have the M phase and cytokinesis occur, you have one division here where one cell becomes two. How do these cells at the end compare to one another? You guys are identical cells. And we have two of them, and are they haploid or diploid? Diploid. They're diploid assuming that the beginning cell was diploid. So for us, it would be 46 chromosomes, and we'd end up with two cells with 46 chromosomes, and we'd have more body cells, so skin cells or whatever it may be. Cytokinesis is not the same for every type of cell. It differs in animals versus plant cells. In animal cells, what do we have? That's what I drew up here. This is the cleavage furrow. So it kind of, like I said, cinches the cell into two. So that's like up here, shown in this picture here. Versus plant cells. Plant cells have, they do have a cell wall, but to make the new cell wall, they form a new cell plate. So let me draw that here. So if this is your plant cell, here are your two nuclei on either side. So we need it to divide to become two different cells. So what happens is vesicles from the Golgi apparatus line up, and then they eventually all fuse together, and that's what's going to eventually form your new cell wall between them. Okay, so that's a little bit different in plants versus animal cells. Another thing I want to point out up here with, let me go back up here to having two identical diploid cells at the end. So for humans, we have 46 chromosomes to two cells with 46 chromosomes. And so that's one of the main things that differs between mitosis and meiosis. Meiosis reduces the chromosome number. So for mitosis, in metaphase is when the chromosomes line up down the center of the cell and the spindles attach, right? And then the reason why they line up single-filely in mitosis is because you want one of every kind of chromosome in your cells at the end. So you want all 46 chromosomes, versus in meiosis, when they first line up, they line up with their pairs, and their pairs separate into different cells so that you only end up with one of every kind of chromosome in the cells. And so that's a major difference with the logistics of mitosis versus meiosis and why you end up with different numbers of chromosomes at the end. Okay, and one last thing with the cell cycle is that not every cell actually goes through the whole cell cycle. So I talked about prokaryotic cells, bacteria. Do you guys remember what they go through? Yeah, these guys go through binary fission, and this is a much simpler process. Why is it much simpler? Bacteria don't have a nucleus, so they don't have to get rid of the nucleus and so on. They only have one chromosome, one big circular chromosome. They don't have to worry about the chromosomes all lining up. So therefore they don't have the spindle either, because they don't have to worry about that organization. And like we have 46 chromosomes lining up, and we have to make sure that each cell gets a copy of every one of those. So it's real simple for them. So binary fission, it's basically the cell makes a copy of its chromosome and divides in two, and then each cell gets a new chromosome. So I'm going to put here, this is some things about this that bacteria cells do not have a nucleus. They have one chromosome and no spindle, and that makes it so that it doesn't need all this other stuff that was going on up here. All right, so basically, as I said, binary fission just makes a copy of the chromosome and divides in half. Okay, so that's chapter 12. Oh, well, just kidding, there's more chapter 12. I forgot about the last section, which is about how we control the cell cycle. So why is it that our body cells don't divide forever and continually divide? Cell cycle control. So a couple of things that are involved. So we need to review the checkpoints. So there were 3 checkpoints that we talked about. And so review those. Basically what they do is they make sure that what was supposed to happen previous to that checkpoint happened correctly. Okay. We looked at some chemicals that are involved, some molecules that are involved in regulating the cell cycle. And so review cyclins and cyclin-dependent kinases, or CDKs. And we talked about a specific one, MPF. And so review that and how that controls the cell cycle. We looked at some external factors like growth factors. So growth factors can trigger a cell to divide. So what does that mean? Tying into cell communication again. So here's a receptor and here's a ligand. That ligand, it can be a growth factor. It binds to the receptor, triggers a signal transduction pathway, which will ultimately lead the cell to divide. All right, and so an example of a growth factor I used in the notes is the platelet-derived growth factor, or PDGF, is an example of that. Another thing that kind of controls how often a cell divides are 2 things: anchorage dependence. You guys remember what this means? The cell needs to be on a surface, right, or anchored. It's called anchorage. Sorry, I'm getting ahead of myself. And then there's density-dependent inhibition. I can't speak in dependent. Can't speak and write at the same time. Density-dependent inhibition. This means that the cells stop dividing when what happens? Yeah, when they're touching other cells from all sides, so when they run out of space or become dense, all right, or crowded, they stop dividing. And so normal cells adhere to these different things, and that's what triggers either a cell to divide or not to divide, are these things, and so it controls that. And then the last thing we talked about with chapter 12 are cells that don't really adhere to these things, and those are cancer cells. So we related this to cancer. So cancer cells, these guys don't, I should say cells, don't adhere to some or all of the above. Oops, okay, so these things here. So for instance, cancer cells don't seem to need to be attached to a surface. They don't stop dividing when they're crowded. That's how a tumor forms and so on. And they seem to bypass checkpoints and divide even though they may have something wrong with them, which is the whole purpose of the checkpoints and so on. So that's cancer cells. And with that, we talked about how the process of transformation, which is how a cell becomes cancerous. It's the process that goes through, so it can be a mutation. We talked about carcinogens, things like chemicals and radiation, things that trigger mutations that can trigger a cell to become cancerous and then lose control of the cell cycle. If a cell becomes goes through transformation, it can begin to divide uncontrollably and form, we talked about benign versus malignant tumors. Benign stays in one spot and doesn't seem to hamper the function of whatever organ or wherever it's at. When it begins to divide and adversely affect whatever organ it's attached to, it begins to become malignant. And malignant tumors can eventually go through metastasis, which is when a cancer cell can detach and go into either the blood or the lymph stream to be taken all over your body, and can come out and attach to another part of your body and begin to divide, and therefore kind of quote unquote spread to another spot. All right, and that is chapter 12. All right, so 13 was all about meiosis. All right, so 13.1, was real short. It gave a general overview of asexual versus sexual reproduction. So just give a little synopsis of that. Sexual reproduction means the offspring have some combination of genes from two different parents. So the offspring are not identical to the parents. All right, and so you get new combinations of genes. Asexual reproduction, that isn't the case. So bacteria reproducing by binary fission would be an example of asexual reproduction, because this one little bacteria, the one cell is the whole organism, just makes a copy of its chromosome, divides, and you end up with two bacteria now. So it's reproduced, you go from 1 to 2, but they're identical, and so therefore that would be asexual reproduction. And that's basically what the first section was about. The second section went through some more vocabulary to tie in. So karyotypes. Karyotypes, remember, are your organization of your chromosomes in pairs, and usually by size from largest to smallest, with the exception of your sex chromosomes are usually in the bottom right-hand corner. And so they put them in pairs, and so you have 2 of every kind of chromosome, 1 set from mom, 1 set from dad. So that brings me to knowing what homologous chromosomes are. So those are the pairs. You have, and this is why we have pairs, because you get 1 set from mom, 1 set from dad. Okay, so when we're talking about chromosomes, there's 2 different kinds of chromosomes. There are autosomes versus sex chromosomes. So autosomes are all your chromosomes that don't determine gender. So in humans, as an example, that would be pairs 1 through 22 in humans. Pairs 1 through 22 have nothing to do with determining sex. The sex chromosomes in humans would be pair 23. And remember, in humans, we have XX or XY. And so, and XY is male, right? Okay. This, a lot of people confuse autosomes and sex chromosomes with, back over here, somatic cells and gametes. So gametes are sex cells, and these are your body cells. Sex chromosomes are with your chromosomes within the cells, okay? So for instance, let me just apply this for a second here. In a human somatic cell, if this is a human somatic cell, how many autosomes do we have and how many sex chromosomes? How many total chromosomes should be in the cell? 46, right? How many sex chromosomes do you have? 2. So that means how many autosomes do you have? 44. All right. And so you see that the somatic cell is the cell. Autosomes and sex chromosomes are talking about the chromosomes within those cells. So then gametes is a sex cell, but gametes also have sex chromosomes and autosomes in them. So in humans, in a gamete, how many autosomes would there be? 22, and only 1 sex chromosome. And that gives you your half, all right, your 23 versus 46, yes.
What was a homolog, I think it said in there?
That's the same as a homologous chromosome, okay, yeah.
Is there going to be questions on the test about the sort of
No, no, no. All right, so and as we go into chapter 14, eventually you're going to know. So the last part of 14, I'm going to add with the other, like we're going to, 15 is about genetics too, so we're going to continue on with 15. So you will, there are some disorders eventually that you'll need to know, but not for, yes, okay. So, but let's look at, let's finish this up here first. Let's deal with the test tomorrow. All right, one step at a time. All right. So another thing with this was I introduced diploid versus haploid in the 2N in N. Okay, so knowing that diploid means that you have 2 sets of chromosomes. Haploid is 1 set of chromosomes, so 1 of every type. And then lastly, it's talked about the sexual human life cycle. And there was a picture that showed that with fertilization and the zygote and so on and so forth, and kind of know the life cycles there. All right, so that's the second section. So the third section of chapter 13 was all about kind of what actually happens during meiosis. So like what the chromosomes actually do. So meiosis, I'm going to put here 1 and 2 because there's meiosis 1 and 2. And we need to know what happens in general in meiosis 1 and 2, and then kind of the flow of the chromosomes as well. So let's just look at meiosis here. Meiosis, we start out with 1 cell and it divides into 2 cells, and that's meiosis 1. Then each one of those 2 cells divide into 2 cells, and that's called the second cell division, and that's meiosis 2. So in general, going from 1 to 2 cells in meiosis, what could we say is the general gist of what happens in meiosis 1? And it has to do with what happens with the chromosomes, what separates. Meiosis 1, what separates into 2 cells with the chromosomes? The homologous pairs. So in meiosis, the cells, I'm going to draw a pair of chromosomes right here. Remember in meiosis, they line up in pairs. That's different than in mitosis where they lined up in single file, because in mitosis, you want one of every kind of chromosome in the cells at the end. Here we only want one of every kind of chromosome in the end. So therefore the homologous pairs separate. So that's the role of meiosis 1. So then meiosis 2, what's the general overview for meiosis 2? What separates? The sister chromatids. So this half goes over here, this half goes over here. Sister chromatids separate. So that's, you need to understand that. If you did, I would write that down if I were you. All right. And so what we end up with is we start out with a diploid cell, and we end up with 4 what? Haploid cells. Are these haploid cells identical? No. Okay, especially if you include crossing over. Then also in meiosis there are phases, and so in meiosis 1 and in meiosis 2 there are the same phases, and they're same as what happened in mitosis. So in meiosis 1, to go from 1 to 2 cells, we go through prophase, metaphase, anaphase, telophase, and then the cytokinesis where it splits into 2, right? And that's all part of meiosis 1. Then each one of these cells to get the 4 cells at the end, go through prophase, metaphase, anaphase, telophase to get to your 4 haploid cells. So the phases are prophase, metaphase, anaphase, and telophase. And I'm put 1 and 2 because those happen 2 times each for the cell division. So kind of just know the flow of the chromosomes and what happens to them, how they line up with their pairs, the spindle attaches to them, they separate, and so on. Yeah, for some reason our book, literally they do, because it's really like a snapshot, like, so prometaphase is really halfway between prophase and metaphase, kind of, so they do that. And I was just trying to keep constant with our textbook. Our textbook doesn't have it in there, but technically they would, all right, but okay. And so and then lastly, comparing and contrasting mitosis versus meiosis. So there was a chart that we looked at, and I've kind of been doing that as we go along here. So mitosis, you end up with starting out with a diploid cell and end up with 2 diploid cells. Here we start out with a diploid cell and end up with 4 haploid cells. The chromosomes line up differently, and so on and so forth. So those are the types of things, knowing the difference between them. And then the last part of chapter 13.4 was about how we increase genetic variation. Genetic variation. So what do we do to increase genetic variation? We talked about independent assortment. So what does this mean? This means, remember that when the chromosomes in metaphase 1, when they line up with their pairs, all down the center of the cell, each pair can line up in 2 different ways, remember? And so, and I assign genes like this, could be, big R could be over here in the little r, and vice versa. And each pair lines up independently of the other pairs. So they line up with their pairs, but they each one have 2 different ways to line up. And so therefore, I introduced the formula 2 to the nth power. 2 meaning that each chromosome pair can line up in 2 different ways, and n is the haploid number for the organism. And so, I remember I talked about how the haploid number for us is 23. So 2 to the 23rd power is 8 million, over 8 million different combinations. And so just based upon how our 23 pairs of chromosomes line up, we can have over 8 million different combinations in our gametes. So that's independent assortment. Throw in crossing over. So review crossing over. Crossing over occurs during prophase. They come together, the chromosomes in tetrads, and cross over and exchange genetic information. So therefore, your cells at the end are different in every time, because crossing over and how it happens is random and occurs differently every time. And then we have random fertilization. So that between 2 individuals, it's random which sperm fertilizes which egg. And so in this case, I introduced kind of to you the rule of multiplication with this, that you know, same two parents, the chance that they have two identical kids that are not twins at two different times. Remember, the male would have to make the same sperm cell twice. Female would have to make the same sperm cell, or egg cell. I don't do this every time. Egg cell twice, and so there's a 1 in over 8 million chance here, 1 in over 8 million chance here. The chance that that sperm is going to fertilize that egg, you take the individual probabilities and you multiply them together. All right, and that's not even considering crossing over. So with all of these things, we get lots of genetic variation and very, very highly unlikely of ever getting the same combination twice in two individuals. Question?
Should we review like the cycles of plant and different organisms?
Be familiar with it. I'm not going to ask detailed questions, but general overview that not all organisms are like us. Okay. Okay, and then lastly, so that's chapter 13, and then the last part is with chapter 14. So chapter 14 is all the genetic stuff, so crosses and so on. So chapter 14, let's see, Mendel. So we need Mendel's crosses. So remember, he had the P1, F1, and F2 generation. Couple of things about the P generation. He said his P generation was true breeding. What's another way to say that? Homozygous, right? So they're homozygous and opposite. They had opposite, the parents, opposite versions of the trait. Of whatever trait. Whatever trait he was talking about. So what does that mean? So an example with the parents could be big R, crossed with little r. So if it was purple versus white flowers, you have a homozygous purple plant with a homozygous white plant. So there you have two different versions of that trait, purple and white, but they're both homozygous. That means that the F1s are always what? Always heterozygous, and showing what trait? And showing the dominant. They're big R, little r. So therefore, the white seemed to disappear in the F1 generation, but seemed to also reappear in the F2 generation. So the F2 generation, how do we get that? We take two of the F1s, and so you cross two of the F1s, and that's where he got that characteristic 3-to-1 ratio, remember? And so the recessive trait kind of reappeared, and that's how he came up with, you know, dominant and recessive. So alleles, the different versions of the gene, different versions of the gene. So example, big R or little r is an example of the two different versions. The laws of, we already talked about independent assortment, but I'll just write it again because it addresses it again in this chapter. And the law of segregation. That means segregation, we've already kind of also talked about it, just didn't coin the term there. That means that when the homologous pairs segregate or separate into different cells, and so therefore you only give one chromosome of each kind to your kids via your egg and your sperm cells. Some terminology, geno and phenotype. Hopefully we know that. Genotype is the gene combination. Phenotype is the physical appearance. With genotype, the genotype can be described as homozygous or heterozygous. And so knowing that, so that could be big R or little r. Heterozygous is big R, little r. If we don't know the dominant gene, the dominant parent, we can perform a test cross. So the test cross, what does that mean? If an individual shows the dominant trait, they could be big R, or big R, little r. So how do you tell if they're big R, or little r? Question mark. Which way? We cross it with what kind of individual? Homozygous recessive. And that way, if you see any kids that are recessive, you know the parent had to be heterozygous. Okay? And then we had crosses. So we did monohybrid crosses, that's focusing on one trait at a time, and dihybrid crosses, so know how to do those. know how to do with those genotype and phenotype ratios. Probability. With probability, there were two rules. Multiplication rule, and which was the other one? Addition. So review those. How do you apply those? We looked at different ways of dominance. We have complete dominance. That's like in all of Mendel's traits. That means like if you have a big R, little r, these guys show the dominant trait. So the big R is completely dominant over the little r. But we also talked about codominance. What's the example I used of this? Examples, AB blood type. Which also part of this, with the monohybrid crosses, you need to know about crossing blood types, all right, like last night's homework. And lastly, incomplete dominance. What's the example of this? Yeah, pink flowers. So this way, the heterozygous, remember, had a red gene and a white gene, and neither red nor white is dominant, but they blend. So this is, you often see a blending, an incomplete dominance. And that's basically it in a nutshell. And we actually finished right on time.